The nested RT-PCR has been developed to detect all Lyssaviruses, including all 16 ICTV-approved and 2 novel Lyssavirus species. The validation of this method has been improved by using over 9,000 animal breed specimens in 10 years of clinical rabies diagnosis and surveillance in China. The nested RT-PCR not only can produce reliable results on fresh breed tissue samples, but also on degraded samples. In addition, this method can be used to assay biological fluid specimens, such as cerebrospinal fluid, saliva, and urine. Thereby, this method can be employed in the automaton diagnosis.
In order to minimize PCR carryover contamination, it is important to prepare negative and positive controls during important case steps. For example, template RNA extraction, mix preparation, PCR setup, and gel electrophoresis should be operated in physically separate areas. It is also recommended to use barrier tapes in RNA extraction and reverse transcription in order to prevent reagents contamination during pipetting. Proper handling, such as preparing reagents on ice, changing gloves regularly, and use of ribonuclease-free materials will prevent the introduction of ribonuclease and eliminate degradation of RNA.
To begin this procedure, extract the RNA from rabies-suspected tissue, skin biopsies, saliva, cerebrospinal fluid, or RABV-infected cell culture as outlined in the text protocol. When ready to proceed, remove the needed reagents from the freezer and keep them on ice.